What are stem cells? Stem cells are a special category of undifferentiated cells capable of self-renewal and differentiation. This means they can divide to produce more stem cells or develop into specific cell types under particular conditions. These qualities make stem cells fundamental in developmental biology and regenerative medicine, as they can replenish or repair damaged tissues. Key characteristics of stem cells. 1. Self-renewal. Stem cells can undergo continuous division while maintaining their undifferentiated state. This ability distinguishes them from somatic cells, which have a limited number of divisions before senescence. 2. Potency. Stem cells have varying degrees of potency, or the ability to differentiate into other cell types. Totipotent stem cells can develop into any cell type, including placental cells. These cells exist only in the earliest stages of embryonic development. Pluripotent stem cells, like those derived from the inner cell mass of blastocysts, can differentiate into nearly all cell types but cannot form a full organism. Multipotent stem cells can produce cell types within a specific lineage, such as hematopoietic stem cells in bone marrow, which give rise to different blood cells. Unipotent stem cells, though rare, can only produce one cell type, but they retain the ability to self-renew. 3. Quiescence and activation. Some adult stem cells, like muscle satellite cells, exist in a quiescent, inactive, state until they are needed, such as in response to injury. Upon activation, they begin to proliferate and differentiate, playing a crucial role in tissue repair and maintenance. Types of stem cells. 1. Embryonic stem cells, ESCs, derived from early-stage embryos, blastocysts. These cells are pluripotent and can form virtually any cell type in the body. ESCs have been used in research to understand early human development and potential treatments for degenerative diseases. 2. Adult stem cells, ASCs, found in specific tissues. Adult stem cells are multipotent and maintain and repair the tissues in which they reside. Examples include hematopoietic stem cells in bone marrow, which produce blood cells, and mesenchymal stem cells, which can form bone, cartilage, and fat. 3. Induced pluripotent stem cells, iPSCs, created by reprogramming adult cells to a pluripotent state. iPSCs are similar to ESCs in their differentiation capabilities. They hold promise for personalized medicine, as they can be derived from a patient's own cells, reducing the risk of immune rejection. 4. Cancer stem cells, a controversial and active area of research. Cancer stem cells are thought to drive tumor growth and metastasis due to their self-renewal properties and resistance to standard treatments. Understanding these cells could lead to more targeted cancer therapies. Differences between stem cells and somatic cells. 1. Specialization. Unlike somatic, differentiated cells, which are specialized for particular functions, e.g., neurons or muscle cells, stem cells are undifferentiated and have the potential to become various cell types. 2. Cell cycle and division. Somatic cells undergo a limited number of divisions, the Hayflick limit, before entering senescence. In contrast, stem cells can divide indefinitely under optimal conditions, allowing for continuous self-renewal. 3. Gene expression and plasticity. Stem cells maintain an open chromatin structure, giving them a more plastic gene expression profile compared to somatic cells. This allows them to respond to specific signals for differentiation more flexibly. Medical applications of stem cells. 1. Bone marrow transplants. Hematopoietic stem cells from bone marrow or umbilical cord blood are used in transplants to treat blood-related diseases, such as leukemia, lymphoma, and aplastic anemia. 2. Regenerative medicine. Pluripotent stem cells, ESCs and iPSCs, offer the potential to generate tissues for repairing damaged organs. For example, neurodegenerative diseases. Research is exploring the use of stem cells to replace neurons lost in conditions like Parkinson's and Alzheimer's. Heart disease. Stem cells are being tested to repair myocardial tissue in patients with heart failure. Diabetes. Pancreatic beta cells derived from stem cells could potentially replace insulin-producing cells in patients with type 1 diabetes. 3. Organ and tissue engineering. By combining stem cells with biomaterials, scientists are developing methods to create functional tissues and even organs for transplantation. Ethical considerations. The use of embryonic stem cells ** raises ethical issues, as obtaining these cells involves the destruction of an embryo. This has led to debates regarding the moral status of embryos. In response, iPSCs provide an ethically favorable alternative, as they do not require embryonic material. However, iPSC research must still address potential risks, such as tumor formation due to their high proliferative capacity. Future directions and research frontiers. 1. Genome editing. Techniques like CRISPR-Cas9 are being applied to stem cells to correct genetic mutations. This could lead to treatments for genetic disorders, such as cystic fibrosis and sickle cell anemia, at the cellular level. 2. Understanding aging and cellular senescence. Studying stem cells and their environment, niche, can provide insights into aging processes and how to potentially reverse cellular senescence. 3. Bioengineering complex organs. Researchers are working to bioengineer organs like the kidney and liver. This involves using stem cells to grow organoids, miniature versions of organs that replicate some of their complex functions. Although still in early stages, these organoids could eventually help address organ shortages. 4. Cancer research. Understanding how stem cells and cancer cells share certain traits, like self-renewal, can improve treatments that target cancer stem cells specifically, potentially reducing recurrence and metastasis. Conclusion: stem cell research represents a transformative frontier in biomedical science. The ability of stem cells to self-renew and differentiate offers unparalleled potential for regenerative medicine, disease modeling, and personalized medicine. By advancing our understanding of how stem cells can be used responsibly and effectively, we are moving closer to groundbreaking treatments that could address some of the most challenging diseases and conditions. However, it is crucial to balance scientific progress with ethical considerations to ensure responsible and beneficial applications for society.